All right, everyone. It's my pleasure to welcome you to this event today, to this kind of RedTalks. The focus today is going to be on the single cell biology of germinal center B cells and germinal centers. We are very lucky because we have two experts with us today. We're going to be part of a discussion. Our first expert is Katia Basso. And Katia, if that's okay, I'll let you introduce yourself briefly to the audience.
Absolutely. Good afternoon, everyone. And I'm Katia Basso. I'm an associate professor at Columbia University. And I'm happy to be here today with Dr. Pierre Milpied and Olivier Elemento to talk with you about single-cell transcriptomics and how this is contributing to our understanding of B cell maturation during the germinal center reaction, as well as lymphomagenesis. And indeed, one of my main interests is really the understanding of the physiology of the germinal center, and in particular of the transcriptional networks that drive this very specific stage in the life of the B cells, and also the lymphoma-associated genetic alterations that can disrupt or hijack these physiologic programs. So I think Pierre can introduce himself.
Yes, thank you, Katia. So my name is Pierre Milpied. I'm a researcher in Marseille, France. I'm in the Center for Immunology of Marseille-Luminy, and I'm affiliated with INSERM also. And I am very interested in B-cell immunology. And in my lab, in general, studies the dynamics of B-cell differentiation or maturation in several contexts. My favorite context to study is the germinal centers. This is the place where everything started for me for studying B-cells. But since I have applied single-cell studies to study germinal centers, also I have been interested in B-cell lymphomas, the ones that are derived from germinal centers. So I started with follicular lymphoma in the lab of Bertrand Nadel in the same research institute. And now I have also interests in the plasticity or the heterogeneity of lymphoma cells in different entities, and to sort of study the things that are in common with physiological immune responses or that are different from physiological immune responses in lymphomas. And we're always very dependent on using single-cell resolve technologies. And now I think we're going to talk about it also, spatial omics, a new field that is starting. So very happy to be here and discuss the topics with you.
Fantastic. Thank you so much, Katia and Pierre. It's great to have you. I'm going to be the moderator of this discussion today. I'm Olivier Elemento. I'm a professor of Cornell medicine, which is the medical school of Cornell University. We are here in Manhattan. And we've done quite a bit of work also in B-cell lymphomas and normal B cells using single-cell techniques, now moving towards spatial technologies. And I'll be happy to also provide some context for all of this. So let's get started. So what we're going to do today is really to try to have a discussion with these two amazing scientists at the cutting edge of technology and B-cell biology. I think we're going to start by maybe trying to get a sense of what kind of technologies are being used right now for single-cell analysis of germinal centers. What are the techniques that are being used? What are the strengths or limitations? Maybe we can try to also cover some of these aspects. So I know that Pierre, you've prepared a little slide deck, just a few slides to give us an overview, and if that's okay, we'd love to hear from you about what is being used and for what.
Absolutely. So yes, I have prepared this that I've taken from a review that we wrote a few years ago. But so the traditional way, which has single-cell resolution to study germinal centers, has been flow cytometry, and we have good surface markers and monoclonal antibodies to identify and purify germinal centers as a whole population with the CD10 or CD38 markers in humans, or GL7 and CD38 negative in the mouse. but also to subset those as light zone and dark zone phenotype based on the CXCR4 and the CD83 marker. So this is the sort of entry point every time. It is still the entry gate for studying germinal center B cells in physiology. But then there's, obviously we want to go deeper, and the field has progressed a lot in the past decade and even more than this by using single cell resolved analysis, because the germinal center is really the place where single clones expand and mutate and differentiate. So really the single cell resolution is very important to understand the dynamics and the rules of affinity maturation. So here there has been ways to track the B cell receptor phylogenies and differentiation and maturation at the single cell level with quite simple tools like single cell RT-PCR and Sanger sequencing, or single cell in vitro expansion, differentiation, and then studying the supernatant or sequencing the cells that have been differentiated from this. And these studies are really important. They're still used nowadays. And what is also has been available to GC studying labs since about 10 years, but more popular in the past 5 years, has been to do integrative single cell RNA sequencing to study everything at the same time. Because with those technologies, starting from a cell suspension, we can isolate the cells of interest and study in the same cell the transcriptome, so the gene markers, study the differentiation trajectories from those gene expression, but also the BCR sequence, so that we can draw a phylogeny and connect the phylogeny to the state of the cells. So if we want to see a little bit how it's been done with human, to study human germinal centers, and we have done some studies in our lab, and Katia has done a wonderful work also in her lab. She'll talk about her results a bit later, I think. So we start from a piece of material, which is a tonsil or lymph node fresh sample, or it can be dissociated. I mean, we need to dissociate into a single cell suspension. And already you can see that there are two limitations to this, is that obviously a sample contains multiple germinal centers, and we isolate the cells from multiple germinal centers. So this is always something that we have to keep in the back of our head, that we're not studying single germinal centers in almost all of our assays. And if we want to study single germinal centers, we will need to have other ways to purify the cells. We take a single cell suspension. So we disrupt the tissue and the spatial information is lost. And this is something I think we'll get back to. There are ways that we can study multiple samples at the same time by barcoding samples and multiplexing them before sorting and capturing them for single cell RNA sequencing. Most of the studies studying germinal center B cells use enrichment, because even though they are quite frequent in inflamed tonsils, they may not be sufficiently frequent, or if we want to really study a lot of cells, we need to enrich them based on phenotype. It's not necessary, but it can be useful. So some studies in tonsils have done whole tonsils sequencing, and then they use only the gene expression to purify the germinal center cells from their transcriptome. And then there's multiple ways to do single cell RNA sequencing. The most popular one is the one that is commercialized by the company called 10x Genomics. I have no share in 10x Genomics, but it's a reality that in most of the labs, we're using that technology, which is based on droplet emulsion to capture cells into tiny droplets with barcoded beads that capture the mRNA. And from those, we can derive single cell gene expression library and single cell B cell receptor libraries. And if we have used barcodes, we have barcode libraries that we pool and sequence all together. So in most labs, the scale of the experiment is to capture around 10,000 cells per run. And you can multiplex several samples in a run, or you can choose to do several runs on a single sample also. There's multiple ways to do it depending on the number of cells that you want. And the sequencing depth is here. So basically we target enough depth to capture, to saturate the library so that we capture all the information that we can find in those single cell RNA-seq which is not the full picture. We have to keep this in mind. There's a sensitivity threshold for single-cell RNA-seq, but it's still good, and it's progressing. So every year or every 2 years, there's a new version which is more sensitive. And this is where the fun begins. This is where you get your data, and now this is really where the tricky part starts. So so the analysis is really key. And I mean, I can go through the major steps of the analysis. This is what I have in the slide here. But if you have, if Katia, you want to jump in at some stage to say something, of course you can feel free. The basic.
I think Katia will give us a good sense of the the application. But yeah, if that's okay, we'd love to hear a little bit, maybe briefly about what are the major steps in the analysis of this kind of data. I'm sure a lot of people are generating this kind of data, and I think if you can give us a sense of what are the major steps, that'd be great.
So the first part is really classic single-cell RNA-seq analysis, quality controls, and this is not specific to germinal center B cells. What is specific to B cells is that they have immunoglobulin genes that they express, and it's an information that is, the gene expression of immunoglobulin genes is obviously important, but the genes, the repertoire, is an information that is in a way orthogonal to the transcriptomics. It's a metadata. It's a data that goes on top of it. It's an extra layer. So you don't want that extra layer to be the main driver for clustering or for trajectory. So it's important to have those genes. Either you can tag them and make sure they're in sort of a separate category for genes, or you can also sum the expression and combine them into a given pseudogene. For every cell will have expression of a pseudogene. And this will be important for the next step of the analysis, where, again, this is classical single-cell RNA-seq, where you want to focus on the main sources of heterogeneity within your data set. So you compute the most variable genes, and you will restrict your analysis to a space where only the most variable genes will be important for segregating or clustering your cells. And we typically focus on 2,000 to 4,000 highly variable genes. It depends on the studies. And this is where we remove the immunoglobulin genes from the highly variable genes, because we don't want those repertoire genes to be the main source of clustering or of clustering when we do dimensionality reduction. So then, starting from the highly variable gene, we perform dimensionality reduction with principal component analysis, and then another layer of dimensionality reduction so that we can represent the data in 2 or sometimes 3 dimensions with so t-SNE, t-stochastic neighbor embedding, was very popular until maybe 5 years ago. And now UMAP is uniform manifold approximation and projection, I think. It's a data visualization technique that relies on summarizing basically the information of 30 to 50 principal components into a 2-dimensional map, where the cells that are in proximity, as you will see in Katia's work, the cells that are in proximity share a transcriptional profile. And then we can cluster the cells in the same space of principal components. And we go to the next step. And the next step is to try and understand better why the cells cluster together and what does it mean and what is the source of that clustering. So there's one thing that jumps to your eyes when you study the germinal center and you do those clustering is cell cycle. As you know, germinal center is the place where B cells proliferate a lot. And so a lot of the cells are actively dividing, and cell cycling in a cell involves a lot of transcriptional changes. A lot of genes are co-expressed and co-evolve during cell cycle. And this is a major source of heterogeneity in the data sets. So it's interesting, but it's not the only thing you want to see. And the problem is that cell cycle is the main thing you see in the first intention. So this is where we have to use tricks to go beyond cell cycle when we analyze the data. And there are mainly 2 tricks. One is bioinformatic or algorithmic trick, is that we regress the impact of cell cycle on the expression of the genes. So this is a mathematical transformation of the data matrix. And the other one is simpler and in a way very robust too, is to separate the cells that are in G1 phase and the ones that are actively cycling, and to analyze those separately. So we subcluster the cells. This is what has been done in Katia's work. And then we take one part after the other, and we study them separately. The things that we can do with single cell analysis is to try and connect the different clusters together, because in the Germinal Center, I mean, there are clusters, but there are also continuums of states. And so we connect them through trajectory. And then we can see, you know, what are the genes that are involved in the trajectory, what is their ontology, which pathway they are coming from, etc. And the extra layer, which is very interesting too, is the BCR layer. And this is, as I was telling you at the beginning, this is orthogonal to the gene expression. This is something that you derive from the same data set, but that you add as an extra layer, and you can then group the cells by isotype, by their expression of a given V gene, or their clonotype based on the VJ or VJ junction sequence, CDR3 sequence. And then you can study whether different clonotypes have different trajectories, whether this could be associated to affinity maturation, to try to see, you know, which subset have presumably more mutation than the other, etc., etc. So all the tools that people have used to study affinity maturation with single cell RT-PCR and Sanger sequencing, you can then repeat and add as an extra layer to the analysis that you've performed before. So this is basically what has been done in the studies that have shown the diversity of B cell types in the Germinal Centers. And it's not specific to Germinal Centers, and you can study B cells, also lymphoma B cells. I mean, this is the way we study them with single cell resolution.
Yeah, that's great. Thank you, Pierre. That's really great to get a good sense of what are the key steps in the analysis, and to hear a bit also about the technology itself. Pierre, there's a question for you in the chat. What I'll do is let you answer the question, because I think you can answer it, while we continue the discussion. A question from Saeed about
so
BCR
Saeed, can you extract somatic hypermutation from the BCR data analysis? Absolutely. So we get full-length sequence of the V of the heavy chain, the V gene, up to the J, even up to the start of the C region in those assays. So you can call the isotype, you have the full sequence that you can then align to the germline and call the mutations for the V, for the heavy chain and for the light chain. So you get somatic hypermutation data. Absolutely.
Cool. Maybe you guys can continue the discussion on the chat if you want. Maybe, I don't know, maybe Saeed will want to hear about specific software or methods that could be used for this. But I just want to make sure that we also hear you, Katia, about your technological choices and the techniques that you use for your single cell analysis. Are these the same type of techniques, or are you using anything different vendors, different software, different steps in the analysis? We'd just love to hear from you before we go into the details about the biology and your findings.
Well, as Pierre mentioned, I think that, again, I don't have any interest in it, but the 10x Genomics is the platform that is more widely used. There are other available. I would say some of the limitations are about the throughput. So 10x Genomics indeed provides a very high throughput, and this droplet technology is extremely efficient. Of course, it comes with a cost. So that is probably something that, at least in a research setting, is still limiting the size of the experiments that we can plan. Regarding the analysis part, I think Pierre outlined very clearly what are the key steps, and I think that we all share about the same pipelines. Technically, there is a wide array, I would say, of tools that are available. There has been a lot of effort from the computational, scientists to develop even what we can call frameworks, right? The kind of integrated frameworks to put together all these different steps in a unique tool. So I can just mention some. Again, I don't want to take away to whatever I don't mention, but I think many people use the SURAT package that is commonly used. There is equally, good approach in the Python environment with Scampi, and I'm sure many people use part of a framework and then integrated with other tools coming from different frameworks, and these absolutely work perfectly. So I think that yes, there are tools that for a starter are there, available, everything in one package, and you can go from your quality control to generate your cluster and analyze your data. But there is also plenty of room to integrate a different portion of this analysis according to what you want to look in more details, taking tools from different directions. Of course, I want to emphasize that we need complementary expertises in this type of work. So it's very relevant to have someone who knows the biology of the system, who has the technical expertise to handle the biological specimens, but is equally essential to have someone with the computational expertise to look into this data, because these are data that can be misleading if they are not, correctly analyzed and interpreted. So I think that this is really a type of experiment where we need to go hand in hand with colleagues with different backgrounds and expertise.
Yeah, that makes a lot of sense. Thank you so much. That's very relevant. I think we are going to move on to maybe the biological part of this talk. We'll come back to the technologies and maybe talk about spatial techniques, because that seems to be all the rage and really the evolution of the field. But maybe let's go into the biological part of this discussion. I think we've learned a lot of things about germinal centers and B cells in general using single cell RNA-seq. And in fact, you, Katia, have published some really amazing studies on single cell analysis of germinal centers. I was hoping that you'll be able to give this audience a sense of what you found. What are the major discoveries that have come up from your papers and maybe from the field using single cell analysis?
Yeah, we were discussing with Pierre when we were, organizing a little bit the structure of this conversation, and we agreed that it was probably useful to show a couple of slides, also to help the discussion, to summarize some of the observation that not only we had, but other researchers in the field actually came up with. So maybe just to put everyone on the same stage, I will summarize what are the key aspects about the germinal center reaction first. So we all know that B cells really start their life in the bone marrow, and it's in the bone marrow that they acquire the ability through VDJ recombination to express a functional B cell receptor. And it's only when they have a functional B cell receptor on the surface that they move out. They go to the secondary lymphoid organs. And here is where we can have these germinal center reactions happening. And how a germinal center reaction starts is when a naive B cells with its a B cell receptor can recognize an antigen, receive stimulation from other cells, in particular T cells, and is practically activated and start to proliferate, and change also his features, becoming what we call a germinal center B cells. The germinal center are really histological structure, are very clear, in a tissue section. And histologically also we can recognize what we call the two compartments of the germinal center, that are the dark and the light zone. These two histologically distinct areas of the germinal center are actually functionally distinct compartments. In the dark zone, germinal center B cells undergo very rapid proliferation, and they do something very dangerous but essential. They mutate the variable region of the immunoglobulin genes. This is what does give variability, additional variability to the B-cell receptors, so the antibodies, and change also the affinity for the antigen that has triggered the reaction. After this dark zone cells migrating the light zone compartment of the germinal center, and it's there that through interaction with a number of other cells types like follicular dendritic cells, different type of T cells, these B cells can check how their B cell receptor has been modified, and if it has acquired features that are useful, unuseful, or dangerous. And practically in this selection stage that occurs in the light zone, B cells can be redirected through different fates. They can be sent back to the light zone through what we call a cyclic reentry mechanism. Indeed, the germinal center cells very often undergo multiple rounds of somatic permutation in the dark zone and selection in the light zone. They can be eliminated by apoptosis. This is actually probably the most common fate for germinal center cells. Or they can be primed to commit to post-germinal center differentiation and become memory B cells or plasma cells. So memory B cells will retain the memory of a good B cell receptor that has been refined during the germinal center reaction. Plasma cells instead will secrete in form of antibodies these refined receptor generated during the germinal center reaction. So overall, the goal of the germinal center, it's really the one to change the affinity of the original B cell receptor to produce a good immunoresponse. The germinal center reaction, as I mentioned, is also the site where B cells undergo genetic changes, physiologic genetic changes like somatic permutation. And also I didn't mention class switch recombination that requires cutting DNA to change the constant region of the B cell receptor and give the opportunity to produce antibodies of different classes. Both these genetic mechanisms have actually an impact also in the generation of unwanted genetic alteration that are part of the malignant transformation process that indeed can affect germinal center B cells. We know that a number of different lymphomas have their cell of origin in germinal center. Here I mentioned the most common, Burkitt lymphoma, follicular lymphoma, and diffuse large B-cell lymphoma. They all share a germinal center origin, but they have differences among themselves. For example, Burkitt lymphoma appears to be closer to a dark zone germinal center B cells, while follicular lymphoma are more related to light zone germinal center B cells. Diffuse large B-cell lymphoma is really the most heterogeneous among the germinal center derived lymphoma, and has been, related to very different cell type inside the germinal center. Of course, the germinal center reaction already at this stage appear to have some heterogeneity, but with the advent of single-cell transcriptomic, we and others, actually, Pierre was, when working with Nadel, was among the first who tried to investigate the transcriptional changes at single-cell level in the germinal center. And then work from us and others have enlarged this picture. And I would like to emphasize that these are only some of the work that have been done. Especially these are focusing on human germinal centers, and with really a preference on the normal side of the question. There is plenty of work that has been done in mouse, and it's absolutely essential to understand better this reaction. And of course, more recently, also work that is done in the tumors that derive from the germinal center B cells. So here I'm just showing some of the data from one of our work, where here on the top left, you have this classic UMAP representation that Pierre was mentioning during his presentation of the technicalities of transcriptomics. And what this UMAP show practically are dots. Each dot is one cell. And the color that you see there are associated to cluster of cells. When we talk about a cluster, means a subset of cells that share significantly their transcriptome data. And what you can see from this UMAP, I think it's pretty obvious that first we can, of course, see dark zone cells and light zone cells. This is what we knew, they are there. But we can appreciate that there is a third large group of cells that here we call intermediate zone. Other people call gray zone. There are many ways to call it, but practically are cells that cannot be called dark or light. They have clearly intermediate features between these two compartments. And they are not a small fraction of the overall germinal center. They represent about 1/3 of the cells in the germinal center. The other observation is that we can also see cells that, although have features of germinal center, I want to emphasize that all the cells you see here are actually sorted based on surface marker of germinal center B cells. But when you look inside at their transcriptome, you can clearly identify subset that already appear to commit to post-germinal center differentiation, specifically plasma cells and memory B cells. So the changes in the transcriptome occur early in the germinal center at the point that can be captured on cells that on the outside are clearly still germinal center B cells. Another point that I think is relevant is that even when we look inside cells on which we put one label, for example, dark zone or light zone, you see on the right, there are actually multiple subpopulations inside this group of cells sharing transcriptional features. And this is actually relevant to point that whatever we call a light zone cells probably is not identical to all the other cells that we call light zone. Same for the dark zone and so on. So there is heterogeneity, and this heterogeneity goes beyond the surface markers and goes really beyond the label that we usually attach to these cells. There is something else that we can do with this type of data. So Pierre was mentioning the ability to have a computational tool that try to infer transition or connection between these different populations. So in this specific example, we were trying to see the connection across this germinal center subpopulation. And you can see I pointed a few of these nodes and separation. You can really capture the re-entry cells that from the light zone start to express marker, in particular MYC, that would redirect them to re-enter in the dark zone. You can capture SIL cells again in the light zone that instead acquire a completely different transcriptional program that commit that to plasma cell. And similarly, you can identify cells that change their program to commit to memory B cells. So these inference algorithms are actually useful to point us to connection and between these different populations to also investigate the programs that would be critical and relevant in priming cells to acquire very specific transcriptional program. So practically, this is really a quick summary, but it's just to say how single-cell transcriptomics, at least in the context of germinal center B cells, has moved our view of this bicompartmental structure into what is in reality a gradient of states, in which cells move across different transcriptional states toward then the acquisition of very specific features that will prime them to post-germinal center differentiation. So I see a lot of questions coming in. I don't know if there is anything that we want to address here at this time, or Olivier, you have...
I think that if you want, what you can do is to take a look at the questions and maybe try to answer them on the chat so that everybody can sort of see your answers. But before we get to this, I have a question. Maybe it's kind of a broad question, but these amazing studies that you've done, do you think that they challenge to some extent our definition of cell type? Because you're finding all these cell populations, right? But would you say that there are cell types, different cell types? So how do we revise maybe our definition of cell types based on this ability to assess individual cells with such detail like this?
Yeah, I think that this is actually a very critical point, and I will be happy also to hear what Pierre has to say after I say what I have to say. So the point is this: we were always being used to think about, you are cell A, and then you may become cell B, and then whatever, right? But in reality, these studies shows that you can go through what we call states that I don't know if this can be identified like through identities, and cells can change right across these different states in a continuous way, but also moving back and forward if you want. I think the cyclic re-entry that you have between dark zone and light zone is a very classic example of what we say when cells can go back and forward across different type of identities. So sometimes I say, we are used to say, well, one thing has one identity. Here doesn't look like. It looks like that you can be some type of entity with multiple identities over time. And this, I think, is an interesting concept, not only in physiology, is something that maybe we can discuss later, but looks to come up also when we look at tumors. In particular, I'm talking about germinal center, there are tumors. But I think it's difficult here to say, well, there are completely different population or cells moving across a range of states before acquiring well-defined identity, right? I would say a memory B cells is a memory B cells, but in the germinal center, you will have cells that move across a different identity multiple times before they commit to acquire a well-defined, life definition, whatever you want to call it. So this is my...
Yeah, that makes sense. I don't know, Pierre, you want to react to this or what kind of problem?
Absolutely. I think the single cell analysis has told us to differentiate between cell type and cell state. And it's not an easy distinction. And so sometimes we're not quite sure, is it a cell type or a cell state? And it's also something we struggle with. And it's also something, when you do at the analytical stage, when you have a data set and you cluster your data set, you have to choose a resolution. And you can do, high resolution clustering, which will generate a lot of clusters from the same data set. Or you can do low resolution clustering, which will give you less clusters. In a way, if you have a data set with different cell types, if you do low resolution, each cell type, B cell, T cell, dendritic cell, monocyte, etc., will be its own cluster, and that would be fine. I mean, that's expected. If you increase the resolution on the same data set, then you will have subsets. And in immunology, we're very used to subsets. We like to cut things into different boxes and boxes of boxes. And with phenotyping, this is what we do. But in single-cell RNA-seq, if you want to cut this, you end up cutting continuums of cells in high-dimensional space that we project in 2D space, but you end up cutting a continuum. And then you have to ask the question, how do I cut a continuum, and why do I cut it? And is it really a distinction between two cell types, or is it just, different versions of the same cell? If I had cut that cell, a few minutes later, a few hours later, would it be in another cluster? So in that case, it's not really a cell type, or you can argue on the definition. So I think this is what we've learned, and in a very dynamic environment like the Germinal Center, this is obvious that the boxes are nice for for teaching, they're nice for understanding the concepts, but when you look at the data, you can see continuums. And of course, there are poles, like there's a dark zone state, which is a pole, but how many cells in a GC are in a canonical dark zone state? Maybe 20%. How many cells in a canonical light zone state? Maybe 20% also. But the rest is in some sort of intermediate state. And that's really what we're learning with single-cell RNA-seq.
Yeah, that makes a lot of sense. I see that Bertrand has a question that's extremely relevant to the discussion here, so maybe we're gonna hopefully try to address it. Bertrand is asking whether we think that transcriptional states are reversible as opposed to classical definition of differentiation stages, and do we think that this is a specific property of lymphoid cells, imprinting tumor plasticity, which would not exist to that extent in other tumor types like lung cancer? Pierre, do you want to start? Do you want to try to address this?
Yeah, I think that's a discussion we've had with Bertrand several times about reversibility or irreversibility. That's true. I think that's the way we can look at this. Like those states, you can imagine this. This is what I was saying. If you look at the same cell a few minutes or a few hours or maybe a few days later, it will be in a different state. So in that case, it's reversible. Whereas if we call a differentiation stage, or we say plasma cells are terminally differentiated, it means they will not change as much. Is it a particular property of lymphoid cells? That's a tricky part. Obviously, you can have a lot of plasticity in stem cells or in progenitor cells, and you can find stem cells in progenitor cells in basically all tissues. So I wouldn't say that's a specific property of lymphoid cells. But what is true is that, in essence, a lot of the lymphoid cells have some sort of stem cell property because they are, in a sort of resting, dormant stage, in a naive stage, at least for adaptive lymphocytes. But when they're triggered, then they will change state and proliferate and give rise to different sorts of progenies and memory, etc. So they're built in a way to be very plastic. Probably more than other cell types, but hard to say.
That's man. Thank you. That's really interesting. Katia, anything to add to this? What's your opinion on whether transcriptional states are reversible in B cells in germinal centers, maybe compared to development?
In germinal center, again, beside those states that we all are familiar with, like proliferation, right? That is a state by definition, and of course is reversible. You proliferate or you stop proliferating. But in a germinal center, again, I think that, yes, there is this feature of states, because you have a dark zone transcriptional program is a specific state and identity, right? That is changed when the cells become a light zone cell, but it can go back, right? This is very well established. So you wear dark zone, you become light zone, you change your transcriptome, your feature, even your surface markers, and then you receive a signal that sends you back to become again a dark zone cell. So that is clearly a change of states that is somehow overlapping also to an identity. So here it's a kind of very undefined, I would say, way of defining state and population. While again, I agree, when there are final state of differentiation, like memory B cells, it's memory, although even a memory B cells then can become a plasma cell, right? So B cells probably are really plastic, as there is a plasticity here that is intrinsic to the identity of the cells themselves. So yes.
Yeah, that's
I think that's key. What you just said is that the plasticity could be a core identity. So a GCB cell is still an identity. We can recognize a GCB cell because the GCB cell can be in one of those states, and you expect the GCB cell to be plastic. And so in a way, the plasticity is part of the identity. This is a way to describe it.
Yeah. I think the classical dogma in development, right, is that let's say cell types or cell maybe, I don't know, specific cell types are kind of locked in with an epigenetic sort of mechanism, right? You have here something that changes not in the genome but in the epigenome to be able to lock in a particular state. I guess this question here could be addressed by looking at both transcriptional states but also epigenetic states. And there are some techniques, right, that in theory will allow you to do so. I don't know if you guys have any experience with this ability to do both single-cell RNA-seq and single-cell ATAC-seq in the same cells. That's maybe one way by which this could be addressed. Anybody is doing this, we'll talk more about what's exciting in the future, but just, as a live question related to this topic, anything relevant to say about this?
Well, I agree. It's actually single-cell ATAC in parallel with the single-cell transcriptomic became available, and this is actually one of the tools that has been added in our hands. So we personally in our group do not have yet data on germinal center B cells of this type, but again, the technology will definitely help to integrate these two levels that, as you mentioned, especially in this type of plasticity, and in this context of plasticity could be definitely informative, because epigenomics, it's a key aspect of germinal center biology in this context. I don't know if Pierre has some direct experience with the technique right now.
Not me, but there's a beautiful paper that's just been out in Immunity by the group of Holger Hein and Iñaki Martin Subero in Barcelona, where they have, a lot of single cell, they call it a transcriptional atlas of the human tonsil. And they have a data set combined ATAC-seq. I mean, it's not, it's integrated, so they did it separately, ATAC-seq and single cell RNA-seq. It's not exactly in the same cells, but they have a very nice analysis, and that includes germinal center. So I encourage everybody to take a look, and the data is available, and they've done a really good job in making everything available. And it's a great resource for those people studying germinal center, but also germinal center malignancies, because it can serve as a reference data set. I put the DOI info in the chat.
That's great. Thank you, Pierre. Well, let's move on a little bit. I mean, stay in the domain of biology, and we heard from Katia the sort of cutting edge discoveries that she made and that others have made in terms of what we learned with single cell technologies. Pierre, in your view, anything striking, anything really important that came out from these technologies that you could tell us about?
Absolutely. I think we're learning about the rules of affinity maturation by combining those technologies with with the more analysis, especially on the B cell receptor. So what Katia has shown, and it's been also shown in the mouse, actually for once, the diversity of cell types has been shown first in the human and after in the mouse. So that's interesting. But it's been observed also in the mouse, and the beauty of the murine models, of the mouse models, is that they can be manipulated in order to try to understand better the dynamics. So this is the view of the germinal center reaction with the the affinity maturation dynamics. And really what those single-cell RNA-seq studies in the mouse and in the human have shown is that at the point of selection, affinity-based selection or cell death, when the B cells interact with the FDCs and then with the TFH, then this is where there's a diversity of cell states after selection that can actually be identified through single-cell transcriptomics. The cells that are poised to go back to the dark zone, the cells that are poised to differentiate to memory cells, and the ones that will differentiate to plasma cells. And those have a particular gene expression, but they also have markers, surface markers in some cases, or transcription factors, which can help study them. And so based on this, there was a big question mark on what are the rules for selection? How does one cell goes back to the dark zone or becomes a memory cell and goes into the plasma cell compartment? And the key concept here was Darwin. You know, it's survival of the fittest. It's a competitive setting, the Germinal Centers, so you should have interclonal, intraclonal competition. And the ones that have higher affinity, they take up more antigen from the follicular dendritic cells with their high affinity BCR. So they process that antigen, present it on their surface to TFH, and because they will present a higher density of peptide and on MHC2 at the surface, they will interact more or more frequently or with more intensity with antigen-specific TFH cells in the light zone, and that would give rise to a stronger selection signal. And what has been observed in some systems with transgenic mouse models is that the highest affinity GCB cells tend to differentiate into plasma cells. The ones with intermediate affinity PCRs tend to go back to the dark zone, and the ones with low affinity or actually non-detectable affinity to the antigen become or may become memory B cells. But now that we have tools, I mean, those concepts are being revisited, and I wanted to cite two recent papers, from one in Cell very recently from the group of Olivier Bannard in Oxford University, and one from Ian Cockburn in Immunity. Actually, I think they were out the same month or something. And here they're using not transgenic models to study affinity, but they study polyclonal germinal center responses to infectious agents. Here to influenza virus for the work of Olivier Bannard, and to malaria parasites for the work of Ian Cockburn. And in those settings, what they could see, which is really very interesting, is that the pre-plasma cells, or the plasma cells that derive from the germinal center, they have a very broad range of affinity, and they may be of quite low affinity, even when there is in the same germinal center other clones that have a very high affinity. And that is very striking, because the concepts that we all have in mind, that, you know, the high affinity cells win the competition and overwhelm the low affinity, well, they're not so true anymore. Probably depends on the settings, probably another interpretation of some models where it actually happens this way, but it does not happen this way all the time. And we know this because, you know, now labs are combining those high throughput single cell RNA sequencing assays that we have described with high throughput production of monoclonal antibodies, or FABs, from those single B cells. So you can use the sequence that you get from single cell RNA-seq, the heavy and light chain paired sequences, to synthesize, to clone or to synthesize recombinant monoclonal antibodies that then you can use, you know, to measure affinity to the antigen. And this is what those two papers have done. And there's other papers that have been out studying the same thing for memory B cells. And this is really a new dimension now of studying the germinal center, with, you know, which is revisiting the concept of Darwinian evolution in the germinal center. So I think people now are proposing that it's not only affinity maturation, it's also diversification. So there's a lot of diversification without gain of affinity that is occurring in the germinal center. In the end, you know, it produces high affinity cells, but not only high affinity cells. And that's still a bit mysterious way it works or how it works. But I think that was striking to me, and reading those papers coming out every month on that topic is very exciting.
Yeah, no, that's great. That's really cool. Very, very interesting stuff. Let me ask you a question. I mean, you guys have offered a very B-cell centric view of germinal centers, and with some really incredible observations. Have we learned anything about the non-B-cell component of germinal centers? The other immune cells that are present there. Anything that we learned that is of interest, important, maybe anything that's come up on your radar, in terms of the non-B-cell component? Maybe Katia, we can start with you if you want.
Okay, can you hear me? Yeah. Okay. There was a little issue with the microphone. So, well, of course, as you say, at least we have a very B-cell centric view, but as I mentioned at the beginning, the germinal center does not occur with B-cell only, right? And indeed, there is a role in all these different other cell components that is key. And there is work from several other groups where the analysis is done, as Pierre mentioned, not just on the purified B cells to enlarge, to put this magnification lens specifically on germinal center subpopulation, but in the overall lymphoid tissue. So one limitation is this. When we disgregate a tissue without any type of purification, we take everything that is in the tissue. The germinal center is only a structure inside the lymph node, right? Of course, there are many germinal centers, but they are a very well-defined compartment. And I'm not aware of studies done on dissecting the germinal center structure to be able to study specifically only, the component of the germinal center itself. So I think in this context, it becomes very relevant what we were barely mentioning earlier, these spatial studies, because beside, behind the identity of these other T follicular helper and dendritic cells and so on that are key player in the germinal center, I think what is very relevant is the interaction between the B cells and all these other players. And these interactions are still not really, very well informed by the single cell transcriptomic done from suspension cells. So the, I would say, piece of information on which we need to work more is trying to recapture, yes, the other player, specifically in the context of the germinal center, but the interaction between B cells and all these other players in the context of the germinal center. So, of course, spatial transcriptomic has been available since a while now, but it's a challenging technique where I think the data interpretation still needs quite a bit of work. And also from a technical point of view, we still are, very good, I would say, in the two-dimensional type of analysis, but we need to go three-dimensional with the germinal center. So that is at least, what I think. And, I'm sure Pierre has more to add. He's on the topic, but this is what I would say.
And we'll cover the evolution of the field and the future technologies. But Pierre, anything that we've learned about the non-B cell components of GCs? I'd love to hear from you about this.
Yeah, I mean, I think Katia has pointed out the limitations, that we're not really sure the cells are, the other cells, the non-GC cells, are within the GC or are surrounding the GC in the tissue. That's because we don't have such good markers for, let's say, GC-TFH or GC FDCs and GC macrophages, etc. But still, there are some studies, mostly in the mouse though, some studies on human TFH and the dynamics of human TFH, and presumably what are GC-TFH and how they may differentiate into T follicular regulatory cells with tracking of the T cell receptor, so the clonal identity of the cell. So there are very exciting works, very exciting studies also on the field of TFH, presumably GC-TFH, although you may know that TFH can be found, throughout the follicles and not necessarily only in the GC. So I agree, spatial resolution will be key to answer those questions. I also fully agree that the 3D, the third dimension, the Z dimension, will be key because, there's only so many things you can catch in a single, slice of tissue. And the methods suffer. I mean, the new methods that are coming out with single cell resolution are really good, really, really good. But you're still, dependent on accurate segmentation to make good prediction or good interpretation of the interaction between cells. Because this is our hands-on experience right now that we're playing with this data, is that it's easy to misassign one transcript or a few transcripts to a neighboring cell. And when it's a cell-specific marker, let's say if you catch MS4A1, CD20 in a cell that has otherwise a transcription identity of T cell, you say, okay, I know it's just misassigned. But when it's not a cell identity gene, when it's, let's say, I don't know, a cell cycling gene or activation gene, a pathway that may be found in B cells or also in T cells, NF-kappa B, where is it coming from? Then it gets tricky. And then, we need strong segmentation tools. We need very good bioinformatics to make sense of the data. And we need 3D segmentation. We need 3D analysis of the tissue. And we're not quite there yet, but the technology is going so fast that, it's only probably a matter of maybe months, years, and we'll have that resolution. And I'm sure it will change our view of the Germinal Centers. I'm 100% sure that we will revisit those drawings with the little arrows in the GCs. We'll see something completely different, probably.
Yeah, that makes a lot of sense. We'll do a deeper dive into these technologies in a few minutes, but I have a question that I think is relevant. We've talked about normal B cells and normal germinal centers. As Katia mentioned, there's a spectrum of malignancies derived from these normal B cells, B cell lymphomas, for example, that are frequent and still some unmet needs in terms of treatments, for example. But my question is really, to the two of you, what have we learned about lymphomas, B cell lymphomas, by learning more about the biology of germinal center B cells, or using some of the same techniques in the context of in the context of lymphomas? We talked about heterogeneity, multiple cell populations. Are these features, these observations, translatable to B cell lymphomas as well? Maybe Katia, you want to tell us what you think about this?
Sure. So actually, Pierre was the first one at single cell level to make the connection or the disconnection between the normal and the tumor, right, with the follicular lymphoma study. In the lymphoma field, there is one very key point, that is this cell of origin discussion that was probably, became very relevant in the 2000s when the LBCL, this very heterogeneous lymphoma, was for the first time using at the time very initial gene expression profiling methods, subdivided in at least two categories, the activated B cell-like and the germinal center B cell-like, that were considered two different stages of the B cell development or differentiation, whatever you want to call it. And the key point there was that these ABC and GCB, the LBCL subtype, not only had different transcriptomes, but they had different prognostic value. So patient in the ABC subgroup display a much worse prognosis compared to patient in the GCB group. So this idea that depending on which type of normal B cell was the origin of the malignant transformation would have an impact also on the tumor itself, and indeed also on the feature of the tumor connected to prognosis, has been really connected to the field of B-cell lymphomas. And so when we talk about in this context cell of origin, I would try to clarify, it's not that we really think that that is the specific cell from which the tumor derived. It's more like that that tumor recapitulates some features that are normally found in a certain stage or state of the life of B cells. So initial study, of course, are always focused on bulk transcriptomic data. So all the literature we have until the last few years was bulk transcriptomic data. And also our study in which we were dissecting the normal germinal center B cells using single cell transcriptomic and identifying all these subpopulation, try to connect that to bulk data of DLBCL samples, right? And even if you look at bulk RNA-seq data and you use this very granular signature that you identified in normal B cells, you can see that indeed the tumors are different. And you can reconcile the fact that, for example, Burkitt lymphoma are associated with dark zone cells. And when you use the single cell transcriptomic data for the different dark zone subpopulation, you can see that they relate maybe to different subpopulation, but they are dark zone. If you look at the diffuse large B-cell lymphoma, you see that some are carrying these fingerprints of dark zone cells, light zone cells, anything, even precursor of memory B cells or precursor of plasma cells. Now, what does it mean? We are just comparing, average of expression data coming from the bulk RNA-seq data in these lymphomas with specific signature associated with very clear states in normal physiology. So probably it just reflects what is the majority of the cells doing in the tumor, right, or expressing in the tumor. What is important is that single cell transcriptomic studies that are coming out, actually there are quite a few already, all point to the fact that the tumors are very heterogeneous. So when you go to look inside these tumors that from a bulk point of view looks like, one feature, one transcriptional program, in reality, that one transcriptional program is the average of a number of different transcriptional programs that are found inside one same tumor. So Pierre also has experience, and they were showing, data recently at the ASH meeting. So clearly, inside one tumor, you have a subpopulation of cells that are expressing transcriptomes that could mimic different states that you can find those in normal cells, but they are coexisting. So you will have in the same tumor cells that may carry, signature that may correlate with plasma cells or with lysosome cells, and they are coexisting. So again, I think that that plasticity that we were discussing that is associated with the germinal center B cell and maybe beyond the germinal center, it's something that is somehow reflected in the tumors that derive from this stage of differentiation. And probably we will need a little bit also more data and to try to understand better the extent of this phenomenon and to understand the meaning, right? We don't really have a clear idea of what's the biological meaning of having all these cells carrying these different states in one same tumor. So Pierre, if you want to elaborate.
I think I agree with everything that you said, that the cell of origin is a good tool for classification. It's a bit of a misleading nomenclature, cell of origin. I agree. I would, if I could, I would use, closest normal cell or closest physiological state, let's say, not really cell of origin, but it doesn't really matter. The thing is, what is key is that when we use those single cell transcriptomics tools on lymphoma, we do see a lot of heterogeneity, and we do see unexpected states that you would think, or at least we thought very naively when we first did single cell analysis on follicular lymphoma, GC derived lymphoma, which was thought to be a light zone derived, or very close, at least in gene expression at the bulk level, to light zone germinal center B cells. And we thought we would either find light zone B cells and maybe different states of light zone B cells, but in the end, what we found is more some cells that look like germinal center B cells, not really light zone actually closer to dark zone, and some cells that look like memory cells, not light zone, memory cells. And we saw a lot of cells that were intermediates, in a sort of in-between states that we couldn't really call, which don't have a lot of specific markers. And we see this a lot. And now we have more experience with more diseases, as Katia was pointing out. We presented, Sandrine Rouland in our group presented, in our collaborative group, because we're working a lot together with the people in Marseille and with the people in France on those topics. That in diffuse large B-cell lymphoma, as Katia was pointing out, we see different states within a single tumor, and some that align with the broad cell of origin, like we see cells with a GC profile in a GCB sample, but we see cells that are not in a GCB state or close to a GCB state, even in the GCB sample. And so we see a lot of things that are quite surprising and don't really fit with the labels that we like to put at the bulk level. Now, the question is, what does it mean? And the first question we're asking is, what's the source? Why do some cells have a specific profile and others another? And one of the questions we ask is it genetic? Could it be subclonal evolution that they end up with, mutations in given genes? And so that's obviously genetic mutations in coding genes or in transcript or promoters, enhancers, could obviously change the transcriptome and change the state of the cell. What we see from just one proxy for subclonal evolution is the BCR, because in those lymphoma, very often you still have occasionally AID that is expressed, so you have ongoing somatic hypermutation. And so when we see, couple the phylogeny, the BCR phylogeny of the tumor cells with the transcriptional heterogeneity, in most cases, we don't see a clear subclonal evolution that imprints transcriptional changes. We actually see all transcriptional states in all branches of phylogenetic evolution. In most cases, obviously, we would find other examples. So for now, at least in follicular lymphoma, it may be different in other lymphomas. What we think is that the main source of those different states is interactions with the environment. That, if you bump into a T cell, you would, exchange, the T cell in the environment has CD40 ligand, has IL-21, maybe IL-4, and this triggers a response in the B cell. It would trigger a response in a normal B cell, but it also triggers a response in a follicular lymphoma or in another lymphoma B cell. And if you really think about it, it's really not a surprise. It's quite expected. And that response is reflected in a different state of the cell. And I think this is a lot of what we see is this. And obviously, there are genetic imprinting is very strong, but what we see is on top of this, is plasticity on top of this. Now that's the origin.
That makes a lot of sense.
Know the correlates, that's a whole different question, what it means for the biology of the tumor globally, whether it will grow, respond to treatment, etc. This is another question.
In fact, actually, Bertrand is asking another relevant question, which is, do we think that there's different cell populations within lymphomas will respond differently to therapy? I presume the answer is we're not sure, but I don't know, maybe you guys have thought about this, or have some expectations. Anything that you could say about this?
We have no straight answer. I mean, there are some works that try to do this with in vitro, treatments and try to see if the states changes. But we may have expectations because some of the states, obviously, the cells that are cycling may be more sensitive to, chemotherapy that triggers the cells that are actively dividing, or the states that express lower levels of CD20, for example, may be less well engaged by anti-CD20 monoclonal antibodies, so maybe less targeted by those drugs. But I think for the moment we have no very strong and no clear answer. But the expectation is yes. Obviously, when you design a drug, you design it to target a specific thing that you would think is the most general thing for the tumor. But we know it doesn't work 100% of the time. Obviously, we all know this. So I think the heterogeneity is part of the cause for resistance or relapse. Yeah.
And what's interesting is that R-CHOP actually works pretty well, right? It's able to cure a lot of patients, from the lymphomas, and presumably that's part of the reason is that you have 5 drugs, that are basically targeting different subpopulations, maybe not intentionally in a way, kind of rational way that you just described, but maybe in the end is a reason why it's so effective. But I don't know, Katia, anything to add on this topic?
No, of course, I think it's something that we have still to fully explore, right? It's not that we have enough information to say. And I think there are actually, states that are even, you can see cells, for example, that have a transcriptome associated with cell cycle proliferation, right? And cells in the same sample, they don't. But one question is, are those cells, that do not have now an active proliferation state always like that, or they are moving into, in between different states? So, because the fact that you may find this subset of cells that appear not proliferating, even in tumors where the proliferation rate is practically 99.9% of the cells, maybe suggests that cells move in between these different programs. So I think again that more data would be helpful to dissect this. But clearly, when we talk about tumor, message that we have now is that even with cells that have the same genetic setting or landscape, you have cells that express a transcriptome in a different way with a very distinct feature, I would say, and that is something that needs to be kept in consideration.
Yeah, that's great. Well, that's wonderful. Great discussion about the biology and of B cells and the significance for B cell lymphomas. If that's okay, let's spend the next few minutes. We have about 10 minutes left to talk about the future. We've talked about what's being done now. I'd love to hear from you guys what you think is really the future of investigations of germinal center B cells. What techniques are being used now. We started talking about spatial technologies, but there's a spectrum of them, and as Pierre mentioned, they're fast evolving, they're fast moving. What are you guys doing now? What are you guys planning to use in the future? What is the what's the state of the art, and how is the field evolving maybe in terms of spatial techniques? Maybe Katia, we can start with you.
So in terms of spatial technique, I think the next, there are two directions. One is physiology. So the normal germinal center clearly will benefit from this technique, because what we mentioned before, this interaction between the normal B cells and all the network of cells surrounding them is really where we will have answer to several of the unanswered questions yet. So we really don't know, how a cell in the light zone decide to prime toward memory or plasma cells, right? So which type of interaction could be more useful in one direction or the other, or do they receive the same signal? Is something else? So this is something really that it's open. The other side is, of course, the tumor context. So we were talking about lymphomas as isolated lymphoma cells, but again, it's not, especially if you think about follicular lymphoma, but also diffuse large B-cell lymphoma. These are not isolated B cells, right? There is a tumor microenvironment component that has clearly an impact on the lymphoma evolution, and most likely also to the response to therapy and so on. So spatial transcriptomic, I think, can help in both directions, the physiology as well as the understanding of the lymphoma biology in this way. The other thing that I'm hopeful for is that all these single cell data that we are acquiring will point also to potential useful markers that can be exploited with simpler technique and can become of common use in a diagnostic setting, or also to guide clinical trials on patient stratification and so on. I think that looking at this heterogeneity inside the tumors and being able to dissect potential informative markers associated, for example, with refractory disease or other features that could be of relevance. This is something we are exploring. And again, I don't necessarily think that would be then essential to use single cell transcriptomic to address this. If a marker has a transcript and ideally has a protein, can be converted in very simple assays like immunohistochemistry based, and even a small fraction of cells in a tumor can be identified efficiently. So I think these are two of the main directions that, at least from our point of view, we are trying to, expand our work on.
Yeah,
Pierre?
that makes a lot of sense. Pierre, what gets you excited these days in terms of technologies, Pierre?
I'm excited about the single cell resolution spatial transcriptomics technologies, but I have one problem with them, is the BCR. We don't get BCR from those because they're based on probes designed on genomes, reference genomes, basically, because you want to use as many probes as possible, but you want them to be as generic as possible, obviously. And they're not targeting the B-cell receptor, and there's no way that you will define a limited set of probes to target the diversity of B-cell receptors, CDR3s, that you can get. So I'm excited about those, and we've started to use those methods to study actually lymphoma before studying germinal center, physiological germinal centers. But we're also trying to use methods that will for the moment, lower resolution, but still, that enable us to track the B-cell receptor spatially. That means we're using, so it doesn't work on FFPE sample because it relies on full-length cDNA amplification and then targeting by PCR or enrichment with probes of B-cell receptor cDNA that has been barcoded with a spatial barcode. So for the moment, the resolution is still not good enough, but there are techniques like slide-seq with a 10 micron resolution, which is close to single cell. There are other techniques like stereo-seq, it's called, which has sub-micromolar resolution, which we are in the process of testing also to see whether we could have the best of both worlds, basically, with the integrative view of transcriptome plus clonotype and phylogenies, but also full transcriptome, or at least a good view of this. And so we are very excited about trying to do this. The B-cell community is very active, but still a small community compared to the whole market of spatial transcriptomics. So it may not be such a big priority for companies which are developing those assays. But we've seen in the past that the technologies have also given a little bit of space to immune sequencing of T cells and of B cells. So I assume that it's going to be covered some way by commercial assays quite soon. And I'm excited about this.
Yeah, that makes sense. That's great. We just have a few minutes, 3 minutes left. Any concluding comments, any remarks, to end this great discussion? Maybe Katia, anything that you want to say to conclude?
Well, I think that we have learned something. We still have to learn quite a bit. Germinal Center has a unique feature that is this kind of plasticity and movement of cells and changes that happen in a very well-defined structure. So gives us information in a focused way, right? Heterogeneity, but at the same time in a localized place. So it helps. So we are in a good setting. So this is a place to start to learn about physiology, and of course from there moving to lymphomas.
Great. What about you, Pierre? Any parting comments?
I think I'll try to conclude and answer the question of Bertrand about artificial intelligence.
Okay.
And because it's a point that we had discussed already. So those tools are generating a lot of data. But it's still, I think, Olivier, you were mentioning this also in our discussion previously. What we think as biologists is big data is not big enough for training good AI. But there are a lot of initiatives to try and leverage some AI tools, the most advanced AI tools, to analyze the data or to make sense of the data. So I think this is going to be an exciting direction in the future, because there has been a lot of data that has been produced already, and there will be even more data because the technologies are increasing in throughput. If you think about spatial transcriptomics, what you get from a spot of 2 millimeter, so we've analyzed TMA with spatial transcriptomics, and in a single spot of 2 millimeter diameter, you get 60,000 cells. If you do a TMA with a lot of tumors, you get a lot of cells. So you get a lot of observations. So I think at some stage, hopefully soon, we will get to the amount of data that is sufficient, or at least interesting enough, to train good AI tools that will help us integrate the data, analyze the data, simulate perturbations, because this is also what we want to do to understand better the rules of those biological systems. So it's going to be very exciting to try and use and master those tools. Obviously, you have technology evolving, you have data analysis evolving, AI evolving everywhere. So I'm not sure this is there's going to be a lot of mess also involved everywhere. So we'll have to be to have very bright minds surrounding us, people who master everything, or at least who can talk to one another in order to make sense of all that mess. But I think this is going to be exciting, and I'm really looking forward to the future of what we will be able to discover with those technologies and the rules. I think we're very close to understanding or to predicting biological systems with such quantitative data. The GC is a very nice playing field for this because it's both molecular, cellular evolution in a tiny environment in a nutshell, and with quite a short time frame for those changes. So it's really a very exciting, object to study.
Fantastic. Well, I have no control over the RedTalk schedule, but it looks to me like a RedTalks focus on AI, would be fun to have, and I'm sure Pierre will be delighted to participate. So I just want to say thank you to the two of you, Katia, Pierre. This has been a fantastic discussion. You guys are amazing. You guys know so much and have so many great ideas, so much information that you're able to provide to this audience about single cell technologies and application to B cells. So I just want to say thank you. I want to say thank you to the audience. We've had some great questions as well. I hope that we've been able to answer most of the questions either during the discussion or through the chat. But thank you for this very interactive session. And so that's it. That concludes the end of RedTalks for today. Thank you, everyone.
Thank you, Olivier. Thank you, everyone. Thank you, Katia.
Thank you, everyone. Bye.